Welcome to Biology at Priyojit, my dear viewers. Today we will explore an important and fascinating topic in genetics, that is extrachromosomal inheritance. In this class, we will understand how certain traits are inherited through cytoplasmic factors rather than nuclear chromosomes. We will discuss maternal effects and see how a mother's genotype influences offspring's phenotype, with classic examples like cell coiling in Limnaea, a mollusk. We will also examine the role of Kappa particles in Paramecium to understand infection like cytoplasmic inheritance and its interaction with nuclear genes. These concepts are very much crucial for your undergraduate, postgraduate session, NEET, GATE and other competitive exams, and they help us appreciate that inheritance is not governed by nuclear genes alone but also by cytoplasmic components that shape development and variation. Let's begin. Introduction. Extrachromosomal inheritance is basically the pattern of inheritance in which certain characters are controlled by genes located outside the nuclear chromosomes. These genes are basically present in the cytoplasm, mainly in mitochondria and chloroplast. Such traits are transmitted predominantly through the maternal parent. It shows non-Mendelian inheritance patterns. So what is the basic concept? Most characters are controlled by nuclear genes and some characters are controlled by cytoplasmic genes present outside the nucleus. What are other names of these extrachromosomal inheritance? It can be named as cytoplasmic inheritance, extranuclear inheritance, extrachromosomal inheritance, organellar inheritance, and non-Mendelian inheritance. Genes involved, it is governed by plasma genes present in chloroplast DNA or cpDNA and mitochondrial DNA or mtDNA. So comparison of nuclear and extranuclear inheritance. Location of genes, chromosomes in nucleus, and extranuclear inheritance are found in cytoplasm, that is in mitochondria and chloroplast. Mode of inheritance, nuclear inheritance follow the Mendelian pattern and extranuclear inheritance follow non-Mendelian pattern. Reciprocal crosses, it shows same results, but extranuclear or cytoplasmic inheritance shows different results. Parental contribution, in case of nuclear inheritance it is equal from both parents, but extranuclear in case of extranuclear or cytoplasmic inheritance it is mostly maternal. Segregation, nuclear in case of nuclear inheritance it is typical Mendelian ratios, but in case of cytoplasmic inheritance it is irregular or non-Mendelian. Gene mapping is easier in nuclear inheritance, but in difficult or limited approach in extranuclear inheritance. Somatic segregation is rare in nuclear inheritance, but it is very common in extranuclear inheritance. So nuclear inheritance examples are eye color, height, etc., but cytoplasmic inheritance examples are leaf color, cell coiling, etc. Key criteria for extrachromosomal inheritance, first of all, reciprocal differences. Reciprocal crosses gives a different F1 results not seen in autosomal nuclear inheritance. Maternal effect, phenotype of offspring depends on mother's genotype due to larger cytoplasmic contribution by ovum. Non-Mendelian segregation, typical Mendelian ratios are not observed. Somatic segregation, it is seen in somatic tissues. Example include variegated leaves, governed by plasma genes which are located in mtDNA, that is mitochondrial DNA or chloroplast DNA. So classes of cytoplasmic inheritance, there is maternal effect, inheritance due to infective particles, and true cytoplasmic or organellar inheritance. Maternal effect definition. Maternal effect is a pattern of inheritance in which the phenotype of the offspring is determined by the genotype of the mother, not by the offspring's own genotype, and it occurs due to gene products like mRNA, proteins, enzymes which are deposited in the egg cytoplasm during oogenesis, and it is considered as a special type of non-Mendelian inheritance. What are the important features of maternal effects? Offspring phenotype depends only on mother's genotype, and reciprocal crosses also differ. Mendelian ratios appear one generation later, and it is common in organisms with cytoplasm dependent early development. First example is the cell coiling in Limnaea peregrina, that is a freshwater cell. Limnaea peregrina. Nature of the trait, dextral is right-handed coiling, which is dominant, denoted by capital D, and sinistral is the left-handed coiling, denoted by recessive, and denoted by small d, and it's a recessive character. So key experimental observation which was proposed or performed by Boycott in 1920s, F1 generation or reciprocal crosses, when dextral female capital D is crossed with sinistral male, that is small d, then all offspring will be dextral, and when sinistral female small d will be when crossed with dextral male capital D, then all offspring will be sinistral. So results contradict Mendelian inheritance. So explanation which was proposed by Alfred Sturtevant, cell coiling is controlled by maternal effect gene, and mother's genotype determines cell coiling. Gene products in egg cytoplasm control early cleavage pattern. This is the image showing the cell coiling pattern in Limnaea peregrina or freshwater snail. F2 generation, in F1 generation genotype will be capital D small d, and expected Mendelian ratio 3 dextral is to 1 sinistral, and observed character they are all are dextral. Reason, all F1 mothers were capital D small d. F2 generation, when F2 female genotypes 1 capital D, 2 capital D small d, and 1 small d. Capital D and capital D small d, they are dextral offspring, and small d are sinistral offspring. So observed ratio are 3 dextral and 1 sinistral. So conclusion of the cell coiling experiment is the classic example of maternal effect inheritance, and offspring phenotype here reflects mother's genotype, and Mendelian ratios appear always one generation later. Second example is the Kappa particles in Paramecium aurelia, which was reported by T.M. Sonneborn in 1938. According to this experiment, basically Kappa particles are cytoplasmic DNA-containing bacteria-like particles found in some strains of Paramecium. They produce a toxin Paramecium, and type of strains, their killer strain, they contain Kappa particle and produces Paramecium, and they are immune to its own toxin. But the sensitive strain lacks the Kappa particles and they are killed by Paramecium. So genetic control of Kappa particles, which is dual control, nuclear control, dominant nuclear gene K required for multiplication. So capital K or capital K small k, that is Kappa particles multiply, and when small k, then Kappa particles disappear. So what is the cytoplasmic control? Kappa particles transmitted through the cytoplasm. So this is the image showing the Kappa particles in Paramecium aurelia and how they are inherited. Types of conjugation: short duration conjugation, which involve exchange of nuclear genes only and there is no cytoplasmic exchange. Killer remains killer and sensitive remains sensitive. So the trait is not purely nuclear. But in case of long duration conjugation, exchange of nuclear genes plus cytoplasmic genes, and both X conjugates become killers, and after autogamy, killer is to sensitive ratio is 1:1, and it proves cytoplasmic inheritance. So important points: Kappa particles do not arise spontaneously. Once lost, they do not reappear, and multiplication always depends on nuclear gene capital K, but inheritance is cytoplasmic. So what is the conclusion? Kappa particle is so infection like cytoplasmic inheritance. There is not true organellar inheritance, and it demonstrates interaction between nuclear genes and cytoplasmic factors. So here is a comparison between cell coiling in freshwater snail and Kappa particles of the Paramecium. So as we conclude, remember that extra chromosomal inheritance highlights the vital role of cytoplasmic factors in heredity beyond the nucleus. Maternal effects demonstrate how a mother's genotype and cytoplasmic contributions can decisively shape offspring phenotype, as clearly seen in cell coiling of Limnaea. The Kappa particles of Paramecium further illustrates how cytoplasmic elements interacting with nuclear genes can influence inheritance in a non-Mendelian manner. So together these examples emphasize that inheritance is not governed by nuclear chromosome alone, but also by cytoplasmic components, enriching our understanding of genetic control, development and evolution. Mastery of these concepts is essential for undergraduate, postgraduate and competitive exams, and for appreciating the complexity of inheritance in living systems. Thank you.